And I'll introduce Nigel. Nigel Adams with Unitec has been conducting this brilliant study on gannets. Started off, we've become involved in the last year and this year, or next 2 seasons in fact. In fact, I want this study to carry on. It's such a fantastic study. You'll enjoy this presentation.
Oh, jeez. I'm in trouble now. Oh, well, actually, Chris has already said what I was going to say initially. So there's a lot of people who've been out helping me in the field as well, and a few of them are in the audience this afternoon. And obviously, they had some help, obviously, in terms of funding from Unitec and Birds New Zealand. And then as I've started this close collaboration with Chris, through the trust and the gift funding as well. Okay, so it's Takapū. So we're concentrating on gannets, obviously, and like the I guess the penguins, they, at least in summer anyway, the gannets have a much more restricted foraging range, and really they're sampling the Gulf as opposed to the wider oceanic areas north of the Gulf. So we'll see. Whoops, here we go. Is that the right way? Whoops, and we jumped one. And we're not going to be able to play the video.
Probably not.
Okay.
It doesn't like videos for some reason. It didn't work before.
Okay, well, I had nice bridge video footage there, don't worry, it's fine. It's fine, I'll just talk to it. So really, we've already seen some nice images of spectacular seabird events, and one of those, of course, is the gannets doing their plunge diving on these fish schools. In the most cases, the fish being brought up by some predatory fish. And normally there, I guess, we're talking about in terms of the numbers, thanks to Peter's work through the Trust, we know actually there are 13,427 gannets, at least there were in November 2017, and that's a complete census, so that's an actual count as opposed to an estimate. And they're spread across the Gulf, as you can see, from the inner Gulf, right, at the most southerly point, 988 or 1,000 odd, up to the gannet colonies just off the Poor Knights. And as we've already heard, of course, gannets, as well as other seabirds, are of course top predators. And they're dependent really on a flow of energy to fuel their very energetic lifestyle that is passed through, I guess, what we know as food webs. These food webs can be perturbated, and clearly those ripple effects will then come through or be expressed in aspects of the seabird biology, in this case the gannet biology. However, there's probably some flexibility in terms of the energy. In other words, depending on a particular impact on a particular prey species, there's potential for the energy to be routed another way through to the top predators, depending on the details of the biology of that particular species. So I was particularly interested really in looking at the diet of the gannets, really as an illustration of how that energy is being routed through the ecosystem and the communities of underlying organisms through to birds, and more particularly as well, some of the underpinning trophic levels in a way that you'll see that I tackled a little way on. And I was particularly interested in looking about whether there were differences between the inner Gulf and the outer Gulf. And then in the last season or so, we've also been then combining the diet studies along with some GPS tracking to see whether gannets, in a sense, spatially separate the Gulf depending on where they breed. I guess in contrast to a few of the other species that we've had a look at today, actually the biology of gannets is pretty well known. We know they're coastal feeders, at least during the breeding season, that they catch fish in dive bombs that may go down to about 2 to 4 meters, or then combining that with actually underwater swimming where they can get a bit deeper, with potentially the maximum depth they can get it to probably till about 10 meters. And there seems to be reasonably successful, 72% of dives being successful. We also, just in terms of the specifics of the coastal foraging, there was a couple of studies done off Farewell Spit and Hawke's Bay, the Cape Kidnappers rather, which really just confirms the coastal foraging. So we're obviously doing it in a particular context in the HaurakiGulf, where the actual physical topography of the coast and the fact that we've got an enclosed water body may have some influence clearly on where they can feed and where they may actually express their efforts. Okay, so where were we focusing? Well, our outer colony happened to be Mahuki, just off Great Barrier, and Horu Rock, just off Waiheke Island. These images are actually taken during Peter's survey. I'm sure you'll hear about that a little bit later on, so thanks for that. Probably a little bit different from the other seabird islands. They tend to be rather exposed and little vegetation, which makes field work during summer a little bit uncomfortable. Whoops. What have I done? What have I done? Okay, that's fine. Okay, so the diet studies. Well, initially we had a look at regurgitations, and there's Spencer there triumphantly holding a regurgitation from a gannet. And the reason it's triumphant is probably one of the largest fish that we've ever, single fish that we've ever recorded from a gannet. It's a jack mackerel of about 35 centimeters and about 530 grams. Anyway, these are some more examples of regurgitations that we get from the gannets. And most of the data that I'm going to explain actually is done based on what we term the frequency of occurrence. In other words, did a gannet encounter a particular species on a particular foraging trip? There are other ways to express diet, but that's the way that this is done. The other thing that we looked at, developing the technique, is we actually looked at fecal samples. And in this particular case, we relied on identifying taxa that had been eaten, hopefully not only by the gannet directly, but also the prey of the gannets. And this is dependent on just matching the sequence of base pairs that you get within these fragments of DNAs from a particular gene and comparing that against a library. And hopefully you can then identify the taxa. And then really this is just a summary of the main regurgitation work based across 3 years, from 2017, as you say, through to 2017 through to 2019 during January. And really it's just indicating, I guess, what we probably knew already, that they're taking mainly these surface shoaling fish and squid. However, the proportions of those can vary across years and across sites. So, for example, in 2017, the carrot diet was characterized particularly by large amounts of squid. So the actual abundance of a particular prey item is indicated by the size of the circle or oval and the size of the arrows that then project to the gannet. So, as we can see, 2017 dominated by squid, with things like anchovy and jack mackerel and sori and at Mahuki, and slightly different at pilchard and blue mackerel and kauai for horu. And the pattern changes from year to year. So there is a bit of variation within the diet. In other words, demonstrating that effectively this energy flow through to gannets can shift, presumably depending on the availability of one species versus the next. Because we now have a really good idea of what the primary prey of gannets is based on regurgitations, we can actually, from the fecal samples, differentiate between what I'm referring to as the primary prey and then the secondary prey. So the primary prey is the species taken directly by the gannets, and it showed the same picture. Fecal samples is not quite measuring the same thing as regurgitations, which really are just reflecting what the diet of the gannet has been in the previous 3 or 4 hours, I guess, whereas the fecal samples will perhaps integrate the diet over a day or so. So we expect there to be differences, but the range of species that they showed was similar. And depending the specific way that we actually dealt with the actual fecal DNA, the relative abundances did change, reflecting the sensitivity of the particular approach. So, for example, using this approach, using a particular primer, the arrow squid gave a signal, but it didn't using the other. But the general idea is they're showing the same things, particularly in terms of the primary prey. However, what you also get is the secondary, what I refer to as the secondary prey. And this is an example of a squid that I've dissected open, the squid being recovered from a gannet. And these are then some images that I've taken of what's in the stomach of the squid. That's mainly packed through with the krill that Chris has already referred to. And we were able to sort of compare horu fecal samples. So I'm dealing obviously with vomit samples and fecal samples. It's just great. So the horu and mahuki, and you can see from the actual shape of those bars that effectively they're the same things. And this is the range of species that we're getting as secondary prey within the fecal sample. So there's a big signal from those krill, there's a signal from southern hake, and then swimming crabs, and then a whole things perhaps of what we wouldn't expect at all: snapper, leatherjack, red guinard, and rocklear. So the question is, well, how are they getting in there? Well, presumably they're getting in there because the prey are eating them, but clearly these are relatively small. You know, an anchovy is only about 6 centimetres, although I said with jack mackerel we can get up to about 30 centimetres. So clearly the primary prey are not taking those as adult fish, so they're not taking 18 kilogram snapper, of course. They're probably taking larval fish and eggs, but no doubt they contain the DNA, and clearly you see then a signal from that within the DNA. So effectively now we've, in a sense, describing these food chains at 2 trophic levels, with, at least in that year 2017, indicating the importance of these euphorsids. And then we've got swimming crabs and then the fish species that I've talked about already. More recently now, we've been combining that with GPS tracking that was, as I said, interested in seeing where gannets were foraging, and particularly whether they were, in a sense, separating the Gulf out. And you've seen the approach used, obviously, with a number of other species. To date, we've just done a preliminary sample. Well, by say to date, I mean in January this year, we did a preliminary sample with a small number of birds. And at the moment, we're in the midst of actually doing a much larger tracking study as we speak. There are actually birds out now from Mahuki, hopefully gathering data points about where they are. Anyway, this is the type of information that we get. Previous speakers have talked about this, and obviously we can get a lot of information about what the birds are doing in terms of behavior. So this is 2 tracks of birds of one bird from Mahuki and one bird from horu, and sort of the general pattern would be a commute out where you've got a relatively high speed and low sinuosity of the track. You then hit a feeding area where the bird is moving relatively slowly, and obviously it shows this high sinuosity. Then there's a resting period generally thrown in there, perhaps some feeding, and then the return commute, which it does, of course, upside down from the feeding area. We can now combine all those tracks together to get a general idea of where these birds are foraging. And these are the localities that we've got from the Mahuki birds in January this year, and associated with it effectively what's a heat diagram. And obviously there's a big concentration around the colony, indicated by that bright yellow spot, with concentrations of foraging effort around the hen and chickens, with patches down to the Mercury Islands for 1 or 2 of the birds. Remember, this is only a few birds at present, but we're certainly going to increase the sample size this summer. And then from horu, and as you can see, there's a big concentration within the Gulf, except for one aberrant bird that went walkabout or fly about, and you can see it went all the way around the other side of the Great Barrier to the northern tip of the Coromandel before coming back. You remember? There's always somebody who throws in. I mean, biology is noisy, but so that's, I guess, not unexpected. So clearly, there is definitely some indication that these birds are separating out the foraging area. So these are just all the points together, the white localities being from birds from horu, and the black ones from Mahuki. One of the ideas that I was testing is sometimes what's referred to as the density-dependent hinterland model, would you believe? Really, that means that effectively the birds are competing for food, and so one of the predictions that arise from that is that birds from smaller colonies should, on average, forage a smaller area of ocean than birds from a larger colony. And the other prediction of that is, as we've said, is that there should be this separation. And it seems to be the case that, so if we look at some measure of central tendency of the foraging ranges, you can see that the foraging ranges of birds from Hauraki are about a third more than from Hauraki, the smaller colony. And I've got some graphs showing that, and as well as, of course, associated with the actual mean length of the flight path is also somewhat larger at Hauraki than it is at Hauraki. Of course, these are only two of the other colonies in the Gulf, which are presumably going to impact on the foraging pattern that we see, but perhaps in some support of that, you can see there's a bit of a hole towards the eastern side of the Coromandel, where, as if you've remembered, or perhaps you don't, from the first slide, that there are some concentrations of gannets there as well. Of course, as I mentioned, we've been collecting some diet samples associated with this. This particular study in January 2019, this diet study was done on a different set of birds than the birds that we had tracked, although it was done simultaneously. And there are some indications that this spatial separation does have a signal in terms of the diet. And in particular, sauri is invariably caught within the samples as a whole, and I think is an indication that these birds are foraging closer to oceanic waters. We just don't get it in the inner Gulf area. And in general as well, the abundance of squid in the birds from Hauraki is generally higher than it is in the inner Gulf. So there's some suggestion that spatial separation has some consistent patterns in terms of the way it reflects in the diet of these birds. Clearly, I drew a nice circle for my mean foraging or my median foraging area around my Hauraki birds, but obviously, clearly, the birds were not distributing themselves randomly within that particular year. They had this interesting sort of northeast-southwest axis, and presumably that reflected something of the local conditions over the 4 or 5 days that we did this tracking series. There may not be anything into it, but at least that did coincide with a period where there was some cooler water lying along that particular axis. Although that said, of course, that didn't extend to the inner Gulf. But I guess what I'm saying is that presumably at a small time scale, there are local things going on that do affect the distribution of the foraging effort within those general areas that they do utilize. And just to bring you right up to date, as I said, we're doing the study right now, so this is done just a couple of days ago. And so, thank goodness, up to now, it's still the idea of this density-dependent hinterland model is still working, in the sense that although we've got two birds that came south, they're still north of this line that sort of went from Whangaroa Peninsula to the top of the Coromandel, that indicated sort of the northern limit of the birds foraging out from Hauraki. What we've been able to add to the tracking this year, what we will be able to, is that we're actually able to do some diet sampling of the particular birds that have been GPS tracked. So you can see the purple bird here that went out from Hauraki through to the Omaha, came back with jack mackerel, as did the one that went around Haturu. And the bird that went up towards the Mokahinas came back with anchovy and pilchard. And in terms of what you see in the actual regurgitation is that quite often this, for want of a word, strategy in which the birds go in close to the coast, they're coming back with larger fish and just one or two individuals, yet the fish that go out into the more open areas are coming back with smaller fish like anchovy and pilchard, and there would be a few, actually potentially up to 100 fish in the case of anchovy. So there seems to be two different strategies that these birds seem to be using. Just one interesting point is that you can actually also use gannets as current meters. So you might wonder what this is. Birds or gannets don't fly at night. So if they're caught out at sea overnight, they just roost on the water. So this is a bird that's presumably drifting with the current, and then at a certain time the current reverses and it goes back the other way. So here's another drift. So that's a bird current meter for you. Okay, so I guess where to from now? Well, as I've just said, we're increasing the scope of the actual radio tracker, or the GPS tracking rather, for this particular summer. And clearly one of the things that you can do if you're combining the diet with the GPS tracking, as well as finding out where they're going, you can also get a clear indication of how much effort they're putting in to that particular foraging trip in terms of time flying or how far they've got to go. So you were able to, in a sense, calibrate the changes in abundance of one species versus the next in terms of the diet with actually a measure of abundance. So in other words, if you get a switch from one species to another, are they actually working harder because the whole abundance of fish is depressed or squid is depressed, or whether it's they actually be able to just prey swap and it's not really an issue in terms of the amount of effort they're having to put into actually getting that fish. So that's one thing that we can start to have a look at as we build on the diet combined with the GPS tracking. And then, of course, we can do a lot of other things which are, I guess, interesting from the bird's perspective, or viewed from the bird's perspective, about what do birds do in terms of finding fish. And we can sort of start testing ideas about, whether it's private information, what's referred to as private information. In other words, they have a rule that says, well, if I've been successful on one trip, then the next trip I'm going to go out to the same area, or indeed public information where they get some information about where birds are coming back from and head out in the same direction. And that's on the basis that you've got a bird that's zigzagging across the ocean, hits the jackpot, comes back with food. It doesn't want to do the same thing. It just comes straight back to the colony to feed the chick. So if birds are watching where birds are coming back for, that may be an indication of where the best place is to head out to get your fish in the first place. Clearly, local enhancement is an important factor, and that may be to do with just watching where other gannets are feeding, but also keying in on these feeding associations that have been talked about before. I guess a little bit divorced from that, but there has been some question, well, we've got this spatial separation of areas, well, how is that maintained? Well, there's some suggestion that effectively there's some cultural information that's actually built into the bird's biology, so that these separated foraging areas just reflect the fact that that particular colony or that particular population of birds have historically fed in that particular area, and that's what maintains the separation. So, as I say, from the bird perspective, at least the bird biology perspective, we can start looking at these more subtle indications of where birds forage. And that's me done. Okay.
Not that romantic work, really. You think working with gannets would be fantastic, but I've seen Nigel with blood pouring off his arms. And,